Hey everybody, thanks for tuning in. For those of you who aren't familiar with NewLeaf and with M-troph technology, I've got Dr. Allison Jack, our technical product lead here today, and she's going to give us kind of an overview of what M-troph technology is that goes into our TerraSym platform and why this is something you should be taking a look at for biologicals on your operation.
Thanks, Erica. So M-trophs, what are they? We coined the term M-troph because Pink-Pigmented Facultative Methylotrophs, or PPFMs, as they're known by scientists, doesn't exactly roll off the tongue. So we coined the term M-troph, which really helps when you're chatting about them. They are really unique microorganisms. They live in close symbiotic association with the plants. They live on plant surfaces, both on leaf surfaces and root surfaces, and some are known to live inside of plants as well. They provide a ton of different benefits in terms of growth promotion, and hopefully we'll see some of that today in terms of translating into early vigor. So M-trophs are unique in that they utilize a nutrient source within the plant system that no other microbe can really utilize. So they metabolize methanol as their carbon source, and that provides them with a unique niche in terms of a food source that only they can access. So something that other microbes that live in association with the plant eat is photosynthetic exudates. So as the plant is fixing carbon and sloughing it off into the rhizosphere and exuding it also into the phyllosphere, other microbes will kind of munch on that carbon source, but there is a little bit of an energy cost to the plant because the plant is actually expending energy to fix that carbon from the atmosphere. And what's unique about M-trophs is they're eating methanol, which is a byproduct of cell growth and cell wall degradation and expansion during cell growth. So it's a unique waste product that does not have any energy cost to the plant and cannot be used by other types of microbes. So unique niche, and they're very tenacious colonizers and live in close association with the plant.
So one thing we talk a lot about, at least from a marketing side, is that these are pink.
Yeah.
These little microbes are actually pink. Can you talk to us about what that means from a scientific perspective? Because it's obviously a lot more than something just being tinted pink in color.
Well, as soon as you bring up the pink, I remember I forgot to put on sunscreen today because that is what the pink is for the M-trophs. The pink is a carotenoid pigment that actually protects the M-trophs from UV radiation. So that's one of the reasons that they are able to so tenaciously colonize the phyllosphere, is that they produce their own UV protectant. So again, other types of biologicals that have been commercialized, occasionally you'll have to add a UV protectant to the formulation, and in this case they make their own, and they're super cute.
And that's awesome. And one thing for those of us who aren't doctors, when Allison's talking about colonizing the phyllosphere, basically what she's saying is M-trophs have this really unique ability to not only colonize in roots like we normally see with biologicals, but they also grow with the plant up beyond that root surface as well. So that's another unique characteristic about M-trophs that we like to talk about, because not only are we focusing on what's happening in the soil, but we see this really powerful microbe moving its way up the plant throughout the growing season as well.
Couple quick questions, and you may have mentioned this, but are methylotrophs soil-based or plant-based microbes?
They live in close association with the plant, so they really are plant symbionts. They have an ability to live free living, but primarily they are plant symbionts.
Awesome. And can you talk a little bit about how we measure whether or not the Methylobacterium technology, our M-troph technology, is working?
Sure. Is working or is present?
Either.
Either. Okay. So as to Erica's point for colonization, we have two ways that we look at colonization of plants by M-trophs. One is via plating. So we have selective media that we can use to grow the microbes, which again, because of the their unique ability to metabolize methanol, you can actually put methanol in a petri dish. Not many other microbes can grow on it because it is that unique nutrient source, and then you can count the pink colonies. So you can say, okay, on this plant, you process it, there were this many pink colonies on this plant. For for our M-trophs that have made it all the way through the product pipeline and out to commercial products, we actually have molecular techniques that we can use to not just say that this M-troph is present at this number, but it is exactly specifically this isolate. So we're able to identify them to isolate level using molecular methods to assess colonization.
So what about when they're in the field? That's very helpful. When they're in the field, what are some of the things from a scientist's perspective that you're looking for as far as plant symptomology that would indicate to you that we've got a really effective M-troph strain?
So from the root digs so far this season, some of the things we're going to be looking for specifically today are better color, increased shoot and leaf biomass, increased root biomass, and also some changes to the root architecture as well. In terms of increased number of fibrous roots for corn, and also we will be looking at development of nodal roots as well today.
Gotcha.
So
and
for
obviously yield.
yes, at the end of the season. So for you farmers out there who are tuned in, if you've got terracem on your crops, go out and look at your stand and your standability. How are those rows looking? Are they looking uniform? Do you see earlier emergence, better vigor? And then go out and dig up those roots and see what's happening below the soil profile.